Okay, so it's just gone 5, and I guess we can start. So I'm going to introduce everybody. So thank you for coming today. My name is Val O'Donnell. I'm based at Cardiff University in the UK. As usual, we're going to be around 45 minutes with 10 to 15 minutes at the end for questions. And I want to thank Phil and John for stepping in today at relatively short notice. I'd like to introduce our speakers. So we have two speakers today. So Phil Hawkins received his BSc in biochemistry from Bristol University and a PhD from University of Birmingham. He did postdoctoral training in SmithKline French research and then joined a molecular neurobiology unit of the MRC in Cambridge. He joined what's now the Abraham Institute in 1990 and became a group leader in 2003. Phil, along with Len Stevens, leads a team at the Abraham Institute investigating physiological functions of phosphoinositide signaling pathways. And I don't need to introduce Phil because his work is world-renowned. And in 2013, he and Len, well, in 2013 for Phil, but also Len, have both been awarded FRS for their research because it generated profound insight into understanding phosphoinositide pathway, which is fundamental to a great diversity of activities inside cells, including growth, metabolism, movement, hormone action, and the generation of immune responses. So their work established that cell surface receptors stimulate a PI3 kinase that generates the membrane phospholipid PIP3, which acts as an intracellular signal to regulate protein kinases PDK1, PKB, and also guanine nucleotide exchange factor PREX. There's many more examples of seminal work that Phil, along with Len, have performed. Too much to go into now in my brief introduction, but I'm sure we're going to hear some of this, and I'm really looking forward to hearing more. Jonathan studied biological chemistry at Leicester and then obtained a PhD in new synthetic methods towards the synthesis of Taxol. He then spent 11 years working in the biotechnology sector around Cambridge before joining Abraham Institute, where he now runs the biological chemistry group. And he provides chemistry and mass spec support to other scientists. Anyway, John's personal research interests include analytical methods to study cell signaling and the chemistry of the extracellular matrix. And I think today we're going to hear a combination of work from both Phil and John talking about how mass spectrometry can facilitate phosphoinositide research. And I'm very looking forward to hearing your talk. So I'm going to hand over to you both now.
Okay, thanks, Val. So I'm Phil, this is Jonathan. And I thought the way we'd organise this is I'd try and give a bit of an overview of some of the projects where Len and I's lab have collaborated with Jonathan and where Jonathan's input has really made a massive contribution to our work. And then I think Jonathan will take over and present a case study on just one project, which I think both of us have found particularly enjoyable. So phosphoinositides, they're a family of phospholipids in eukaryotes and present them here in cartoon format, sort of beloved by biologists and hated by chemists like Jonathan. But there are 8 phosphoinositides in eukaryotic cells. There's the parental lipid PI, and this has a typical glycerol hydrocarbon backbone and a 6-carbon inositol head group. And then there's PI3P, PI4P, and PI5P that carry monosterified phosphate groups in the 3, 4, and 5 positions respectively. And then there are 3 bis-phosphorylated phosphoinositides, PI35P2, 34P2, and PI45P2, and then 1 tris-phosphorylated phosphoinositide, PI345P3, which most of us just call PIP3. Nearly all eukaryotic cells have numerous phosphoinositides present in their subcellular membrane compartments, just even under basal conditions. And then these are all interconverted rapidly to one another by a whole series of selective kinases and phosphomonoesterases. And they play a myriad of functions in cells, defining organelle identity and controlling trafficking between different membrane compartments. The PI45P2 in the plasma membrane is also the substrate of 2 major signal transduction pathways. So the first of these is receptor activation of the PLC, which cleaves PI45P2 to produce diacylglycerol and inositol trisphosphate, IP3. And these are second messengers that activate protein kinase C and an intracellular calcium signaling cascade, respectively. We now know there's a massive family of cell surface receptors, all working through different signal transduction mechanisms that stimulate a PI3 kinase in cells. And this PI3 kinase phosphorylates the 3 position on the PI45P2 to produce PI345P3 or PIP3. This PIP3 can then be dephosphorylated by a phosphatase called P10 that takes off the phosphate in the 3 position, or by 1 or more 5-phosphatases to produce a lagged production of PI34P2. And it's PIP3 and PI34P2 that are the second messengers that are generated by this signaling pathway. And the PIP3 and PI34P2 act as messengers by engaging their phosphorylated head groups with a variety of different protein effectors in the cytosol and on the plasma membrane interface with the cytosol in target cells. And there are probably 20 to 30 different effectors that are seeing the PIP3 and PI34P2 that is generated on cell surface receptor activation, 20 or 30 different protein classes in any one eukaryotic cell. But probably the most famous effector that most people talk about is a protein kinase called PKB. And PKB, once it's activated by binding to PIP3 and PI34P2, is then phosphorylates a range of downstream effectors. And probably the most famous of these is regulating the activity of the mTORC1 complex, which we all know is a central integrator of cell growth. So this pathway is used by just about all growth factor receptors to control cell growth, and it's heavily mutated in cancer, which is why it's attracted a great deal of interest over the years. So our collaboration with Jonathan really started supported by the late Mike Wakeland when he became director of the Bainham Institute, in trying to find a way to measure PIP3 by mass spectrometry. So why did we want to do this? Well, our methods at the time were all based on radiolabeling, usually with P32 labeled inorganic phosphate. And those methods required large amounts of radioactivity. We then deacylated the lipids and separated the now water-soluble head groups by strong anion exchange HPLC. And of course, this method is useless if you want to start measuring PIP3 in biopsies, like human material in clinical trials, or just trying to find out what's happening to PIP3 levels in vivo in something like a mouse model. And also, quite honestly, the amount of radioactivity me and Len were handling in those days, I'm amazed we're still here, to be absolutely honest. So we really wanted a way around this, and in particular to measure PIP3 in vivo. Some other people were trying to get around this problem by trying to measure the activation of PI3 kinase indirectly by looking at the activation of the PIP3 effectors, for example, the phosphorylation of PKB. But there were various bits of evidence at the time that were suggesting that, this was not a linear relationship between the phosphorylation of PKB and PI3 kinase activity. And then, of course, there are various other benefits that we all know about to having a really good mass spec assay. And I think the major stumbling blocks to achieving this at the time were like the low stability and sensitivity of the approaches that were being used to measure the more highly phosphorylated phosphoinositides like PIP2 and PIP3. And so this is why we turned to Jonathan, who solved the problem. And I think in part this came from his organic chemistry background, synthesizing these lipids. And he solved the problem fundamentally by derivatizing the monoesterified phosphate groups on the inositol ring with TMS diazomethane. And this gave you a much more useful yield of ions in the ESI mass spectra. So this solved like the sensitivity problem that people were facing. And then to get good selectivity in the measurements, he had adopted his faithful triple quad, and he fragmented the molecules such that you liberated a head group that was methylated, and in parallel measured the positively charged acylglycerol fragment. So measuring this mass pair was what gave the selectivity for the assay. And the method basically worked really well, really robustly with high sensitivity. And of course, this doesn't just measure PIP3, it can measure PIP2 and PIPs by just dialing in the appropriate mass for the loss, the neutral derivatized head group. One caveat in the method is because you're just measuring the mass, it doesn't discriminate between the different positional isomers. And this is something that Takahiko Sasaki's lab, I think, has made some major developments in recent years by tinkering with the LC separation before it goes into the mass spec. So just to give you some examples of how this method's really helped us, first of all, measuring, it's really good for measuring PIP3 in tissue biopsies, which is something we just couldn't do with the radioactive methods previously. And this is just an example of a study we published very recently where we were trying to understand what activates or regulates PI3 kinase activity in a mouse model of prostate cancer. So this model has conditional loss of the PIP3 phosphatase P10 in the prostate luminal epithelial cells. And you can see that this massively raises the PIP3 levels in the prostate tissue after about 12 weeks. And in here, I just show a snapshot where deleting an adapter protein that was really poorly characterized, but we knew was bound to the regulatory subunit of PI3 kinase in the prostate, deletion of PLEC-HS1 causes a large reduction in PIP3 activity in the prostate. And we also went on to show that deleting PLEC actually stops the adenocarcinoma that's generated by the loss of P10 in this model. And this is just an example of the sort of PIP3 measurements you can make in vivo that we just couldn't do previously with the radioactive method. Another little example is just where we were trying to dissect out the regulatory inputs into PI3 kinase downstream GPCRs. So this was a study where we were particularly interested in how GPCRs activating the heterotrimeric G proteins of the GI-GO family in primary mouse neutrophils were able to activate PI3 kinase gamma. And we'd got clues from in vitro biochemistry and some structural studies, largely in a collaboration with Roger Williams's lab, that the G beta gamma subunits from the activated G protein could bind to the P101 regulatory subunit, but also the P110 gamma catalytic subunit of PI3 kinase gamma. But we had no in vivo proof of how important those interactions were. So following on from this work, we were able to genetically engineer mice with knockout or knock-in mutations that we thought would block G beta gamma interaction with PI3 kinase gamma. And this is just an example of the sort of data we got from stimulating the primary neutrophils derived from these mice with an agonist formulated peptides that works through a GI coupled GPCR in this system and produces a nice PIP3 response that peaks at about 10 seconds in the wild type here in blue. And we can show that this is all generated by the PI3 kinase gamma isoform because in the catalytic site knockout, PIP3 response is massively reduced. And then we can dissect out the individual contributions of G beta gamma binding to the P101 or the P110 gamma subunit can make to this PIP3 response. So it's just an example of the quality of the data you can get using this assay and how you can dissect out using a direct readout PI3 kinase activity, how the enzyme's actually being regulated. And now I just want to turn to one other thing that I don't think we'd really thought of when we first turned to this mass spec method. So originally designed it to measure PIP3 sort of in vivo. But of course, now we're measuring the mass of these lipids, we're measuring the whole molecule. Whereas previously we were chopping off the acyl chains and then just measuring the head groups. And then suddenly measuring the mass of the whole molecule, well, that's starting to give us information on what particular molecular species the phosphoinositides were actually present. And we ended up with our first experiments looking at primary tissue, mostly from mouse and human sources. We rediscovered what Brian Holub and his colleagues had discovered in the 1970s with PI at least. But compared to other major phospholipid classes, phosphoinositides tend to be characteristically more molecularly homogeneous, and they were enriched in a particular species with a sterile group in SN1 and an arachidonyl group in SN2, abbreviated C38-4 here for the total number of carbons and double bonds. And we thought this was interesting, you know, because it's never really been looked at. Why would cells care about what molecular species of phosphoinositides they were making? Because all of the work on phosphoinositide signaling had focused on the head group recognition by effectors. Nobody really cared about the hydrophobic chains. And so the first step really was, well, just how common is this? And I should say as an aside that everywhere we've looked in a particular cell context, the PI, the PIP, the PIP2 and the PIP3 have very similar acyl chain compositions, which is what you'd expect if they're all rapidly interconverted amongst one another. But because our methods aren't distinguishing between the different regioisomers, you know, there's still some possibility that some of these might be different, and that's something that would need to be looked at carefully. So, you know, how common is this? So this is something that Vishu and Jonathan went about trying to find out. So first of all, here are some neutral loss scans where we're just looking at all of the diacylglycerols that are attached to an inositol head group. And here's an example from mouse liver where we can see they're enriched in the C38-4 isoform. That's what we expected to see. But of course, biology is never as simple as you'd hope it is. And so here's a snapshot of looking at some other organisms. There's worms, flies, and yeast, where you can see the situation isn't as simple. In worms, interestingly enough, they're quite homogeneous, but with a different acyl chain, and even more unsaturated combination, C38-5. The flies and the yeast are more heterogeneous. And then in slime molds, we had a real shock, and they're amazingly molecularly homogeneous. But it was for a species that we'd never seen before. It's actually not even a phosphatidyl inositol. It's a plasmanyl inositol that contains an ether-linked saturated chain in SN1. And this is the project that Jonathan will speak in more detail about in a minute. Okay, so that looks pretty confusing. And then if we actually looked at a few different tissues in a single organism, for example, in the mouse, again, most tissues are very highly enriched in 38-4, and the next most abundant, the close relative here, 38-3. Here in red and horrible pinky color. However, it's not universal. We can find some other organs where that enrichment is not so clear, and some where it's really, really different, like in the testis. They're really enriched in a C32-0 species. And in some other tissues, prostates here, because we were interested in the mouse prostate model of cancer, they seem to have a lot more of the C34-1. So again, this is really confusing. Cells seem to care about what molecular species of phosphoinositide they have in them. Otherwise, they wouldn't be enriching to this level. But in some places that breaks down. And we still don't have a full understanding at all of what the biological significance of this is. And then if we actually look at some of the cells we culture in vitro, and most of us are doing our biological experiments with, I would say over the years, we found that they're characteristically much more heterogeneous in acell chain composition than the primary tissues. Here's just some examples where these are primary mouse macrophages enriched in C38-4. There's the HEC293s, which is an example that does enrich for 38-4, but here are a variety of breast epithelial lines, some of which clearly not enriching for 38-4, and these are all grown in exactly the same serum in parallel. So there must be some genetic basis for this discrepancy here. It's not just what nutritional precursors, fatty acids, are present in the medium. There's a combination of that plus genetics that are defining the acell chain composition. So again, this was really confusing, and we set about trying to understand how tissues defined what the acell chain composition of the phosphoinositides were. And here, Jonathan really helped us by devising some isotopolog tracing strategies, where we started to look at how the PIs were made and how the different molecular species were created. And we ended up devising 3 types of isotopolog tracing methods. The first of these is tracing C13 nuclei added as a heavy labeled glucose through into the 3 carbons in the glycerol backbone of PI. So this is really tracing the initial synthesis of PI that's made totally from scratch, de novo. So this is from the glycerol 3-phosphate derived via glycolysis, is acylation to form phosphatidic acid, conversion to CDP diglyceride and into PI. So if we look at the initial rate of inclusion of these 3 C13 nuclei into the glycerol backbone, we're really measuring PI that's initially made de novo. However, the PI can also be made from PA and diacylglycerol that come from pre-existing precursors, either from the PIP2, for example, by the production of DG by PLC, and also by the interconversion with the other major biosynthetic lipid pathways in the cell, diacylglycerol and PA, and major intermediates in lipid biosynthesis. So the most obvious way we thought of doing this was to look at the incorporation of inositol. So this should signal the rate of PI synthesis wherever the precursors came from, whether they're made de novo or they're recycled. And so Jonathan made a heavy isotope version of inositol to let us do this. One slight caveat is a few studies had shown that the PI synthase enzyme can actually work in reverse in vitro. So we were slightly worried that some of this inositol might go in through the so-called head group exchange reaction and not report net synthesis. So to look at this, we also devised a strategy where we looked at the incorporation of O18 nuclei from heavy water into the diester phosphate groups of PI. And this is a really neat method that I don't think has been as much used trawling through the literature as I think it might be, because this would seem to us to be a way of looking at phosphate incorporation by mass spec that's really rather analogous to looking at P32 inorganic phosphate labeling in earlier radio tracing studies. So basically, the heavy water, the O18 nuclei, within seconds start equilibrating via phosphotransferase reactions with the nucleotide pool, and then get incorporated into the phosphate groups of various metabolites. And here, the 2 exchangeable phosphates on the diester phosphate are labeled here. And so this reports all PI that's being made from PA that either got its phosphate put on through glycolysis or via the DG kinase phosphorylation of diacylglycerol. And just as a snapshot of the sort of data you can get here, all of this is published, so you can go and look at the full story. But we're just looking at HEC293 cells here, labeling with the glucose, the inositol, and the water, and you can looking at all the different species of PI that are being labeled within these experiments, and you can see that the patterns are really quite different. The original steady state levels of the unlabeled lipids are here on the right, and you can see, as I said before, with HEC293 cells, they enrich in the 38,4 species to quite a high level. And looking at the rate of phosphate incorporation, it pretty much identically matches the different steady state distributions of these species. So all the new PIs made in total, roughly in the proportion that they end up with being at steady state. And something very similar, labeling with inositol. Tracking de novo synthesis from glucose, however, is quite different. And you start seeing the early labeling of species of PI that are really quite poorly represented at steady state. Now, we've cross-calibrated these three methods because each of the isotope log precursors are present at different relative concentrations in their precursor pools. And roughly speaking, this is reporting like 10-fold less synthesis than you're seeing here. So basically, this pattern here is buried within the labeling patterns you see here for total PI synthesis. This type of data are also looking at PA synthesis in very analogous experiments, doing some genetic manipulation, looking at fractional turnover rates of the pools, eventually led to a model which I'll present here, which I hope, well, I'm pretty sure it's correct, it'll stand the test of time, where most PI in most cells that we've looked at is actually made from pre-existing DG and PA precursors. This is the so-called PI cycle, where PLC catalyzed cleavage of PIP2 to produce diacylglycerol, which is stimulated by receptors, but is also going on at a certain rate in basal cells. This DG and PA is recycled back to make PI. And we have evidence that actually this cycle is relatively closed, with relatively poor mixing of the DG and PA with the other DG and PA pools that are used to make the other phospholipids in the cell. And we have evidence that, and I'm sure that part of the organization of this cycle that efficiently recycles the backbone of phosphoinositide back into phosphoinositides is down to the spatial organization of the enzymes that are involved. But we have evidence that the acyl chain selectivity contributes to that selectivity. So C38,4-diacylglycerol that's made on PLC cleavage goes back to PI in preference to disappearing into any of the other major phospholipid pools. And this could be one of the raison d'etres for having an enriched phosphoinositide acyl chain backbone, i.e., it acts as some sort of barcode for the backbone that tells the cell from whence it was derived and routes it back into PI and doesn't let it go somewhere else. And then this highly active PI cycle that's whizzing around like this is drip fed by a de novo synthesis pathway where you're initially making phosphatidic acids that characteristically have more saturated, shorter chain versions. These then get converted into a PI, which is then acyl chain remodeled through the Land cycle to create the particular acyl chain complement that cell has, which is usually 38,4 in most mammalian tissues. So I think at this point, I'm going to exit stage right. Pass you on to Jonathan.
Okay. Right, so we were approached by Rob Kay at the LMB and asked whether we could measure phosphoinositides in dictyostelium. And at the time, there's the issue that when people had tried to analyze them using the traditional methods, they couldn't actually detect the phosphoinositides because the original methods rely on a deacylation. And as we now know, you can't deacylate an ether position, and so what you end up with is quite different. So we had a look by mass spec initially to see what we could see, and it was quite clear very, very quickly that we had something a bit odd. We had a structure which was not the sort of mass that you would get if you had two acyl groups in the molecule, and the fragmentation was a bit strange. Very quickly generated an idea of what might be present, and we thought it was highly likely there would be structure 2 on this side, whereas normally you would find structure 1 in mammalian cells, for example. As you've seen in Phil's section of the presentation, the pool of phosphoinositides is actually very, very clean. It is predominantly one species that's being routed round. So we came up with a scheme for the analysis of the phospholipids. What we wanted to do was initially confirm whether we had an ether or a vinyl ether. So we made the palmitol with 2 deuteriums at this position. The idea being that if it went in as an alkyl ether, then you'd see 2 deuteriums, but if it went in as a vinyl ether, you'd lose one of the deuteriums in that process. We then also wanted to know where the double bond was in the fatty acid chain. We wanted to confirm that. And it was clear from the molecular weights that we had that it was likely to have a double bond in one of the chains, and that it was going to be this chain. And then finally, we wanted to confirm the position of the fatty acid, whether it was at the SM1 or SM2 position. And we thought that we'd do that using PLA2. So when we fed the labeled palmitol, it was quite clear that we were getting 2 deuteriums in at the ether linkage. And we did a time course and a range of concentrations, and we could see the 2 deuterium signal increasing and the unlabeled signal decreasing nicely. And we couldn't see any evidence for the single labeled material increasing at all. So the next step was to identify where the double bond was, and we decided to do this through ozonolysis. At the time, we didn't have an ozone generator, and so we knocked together a Heath Robinson piece of kit by stripping down the platinum electrodes from the gel tank, sticking them into some dilute sulfuric acid, connecting them up to a power pack, and collecting the gas over the anode in a flow of nitrogen and bubbling that into our lipid solution at -78 and doing a reductive workup. So what we got was a full-length molecule and very clearly got the cleaved product, which allowed us to identify the exact position of the double bond in the molecule. The fragmentation pattern was interesting. We saw an increase of 2 mass units in this signal on labeling and this signal on labeling. And overall, based on knowledge of other phosphoinositides and the sort of fragments that we were getting, we thought it highly likely that the fatty acid was in the SN2 position. So we then went on to do the PLA2 assay, and we clearly saw the cleavage of product over time, generation of product rather, and cleavage of the fatty acid off under these conditions. And so to us, that confirms that the fatty acid was in the SN2 position. So in the work that we've carried out at Vabrum, we've spent a lot of time making various materials, both labeled and unlabeled. So here you can see versions of the inositol that we've made with deuterium and O18 in at this position. And we've also made C13 versions of inositol. And we make both of these through a mercury catalyzed ring closure. And if you do that mercury catalyzed ring closure in the presence of O18 water, you get the O18 here as a carbonyl. And then you can do a selective reduction with deuterium to get the deuterium in the axial position here. And we also make labeled fatty acids of various descriptions and incorporate those into the phosphoinositides that we make as standards. And the reason we do all of this is that if you're doing quantitative work, you've really got to understand the response of the mass spec to the compounds that you're putting into it. And what you can see here is a range of PIs that we've made synthetically. We've checked the concentration of these standards very carefully through a number of different methods. And then we've done various concentration calibration curves. And what you can see here, for instance, is that you've got 38.4 coming in with this sort of gradient, and you've got the 34.1 much steeper. And then you down here, you've got, say, a 34.2. And obviously, if you're trying to make statements about the amount of material being converted from one thing to another during recycling, for instance, then you need to know the relationship of the response between the different species that you're dealing with. So in terms of the methylation method that we use, what are the most common practical issues that we've had? Well, the concentration of TMS diazomethane that you buy from various suppliers and even from one supplier can vary from batch to batch hugely. It always says 2 millimolar on the bottle, but actually when you assay it, it can be as much as 4 or 5 molar. Sorry, did I say millimolar? I meant the concentration on the label is often 2 molar, and it can actually be as high as 4 or 5 molar. And so you need to actually find a batch that is at the concentration you want and always order that batch, or you need to adjust the method to take into account a higher concentration. Another problem that comes up quite a lot is people using old batches of chloroform. They've been sitting around for a while. The chloroform is stabilized with amylene, and that amylene will disappear over time. It'll do its job, keep the chloroform in an inactive state. But once the amylene is gone, you can get carbene formation relatively easily in comparison to the levels of the fatty acids that you're putting into the chloroform extraction mix. And so we have had a number of times situations where the arachidonic acid containing species have disappeared because there's been carbene formation in the chloroform while handling on the bench. This really surprised me, but it does seem to be a problem. If you put amylene back into the bottle, or if you use a new bottle of chloroform, your arachidonate species suddenly appear again. And of course, there's also the obvious one where people do stupid things. Column variability is the final issue that we've had. So this is a demonstration of 4 columns bought from the same manufacturer. They're supposed to be identical, but when we've run a PIP3 standard through them, we've had quite different responses. So before we start using a new HPLC column, we always profile it to see if it actually is going to behave the way we want it to. And so that, I think, is all from us. This is upside is to thank everybody who's taken part in the work with us. And thank you for listening.
Thank
Yeah,
you,
thank you.
Phil and John. That was great. I've got several questions, so we'll start with those. First thing was, yeah, it's quite interesting talking about the regioisomers and the fact that you still can't tell them apart. Have you thought of using UVPD or ion mobility? Because you talked about chromatography being used, but I'm talking about new mass spec modalities that are available that might be able to give you information on this.
Well, Takahito has actually come up with a chiral column method which separates them. When we've separated them ourselves, what we actually do is use ozonolysis. And once you've chopped the arachidonate chain down, you can get separation, but then you lose the information about which fatty acids are present. But my view at the minute is that mass spec sensitivity needs to increase somewhat, because if you want to look at the natural abundance of some of these species in primary tissue, I don't think we actually have the sensitivity yet on the mass specs to do that.
Yeah. Yeah, I mean, these would be methods where you actually look, in the mass spectrometer itself, you can get that information from additional fragmentation or from movement on ion mobility. Other ways to do it. Yeah, it just would be interesting. So they maybe haven't been tried yet for this. A question from you, Ed Dennis.
Phil, that was a beautiful talk and great overview on the phosphoinositides. I wanted to hone in on something toward the end of your presentation, which was the difference between PLC versus PLA2 in the remodeling and the consequences. And was not clear whether you can differentiate in what you presented between the contribution of PLA2 and PLC, both of which have the potential of favoring in the pathways the arachidonic being in the 2 position.
There's clearly selectivity in the PI cycle as defined by PLC activation, because you can track the bolus of extra 38,4-diacylglycerol phosphatidic acid and how it goes back into PI. You can track that because there's a dynamic increase in it when you stimulate the PLC, so it's PLC generated, and you can block that pathway with DG kinase inhibitors that stop the DG being converted to PA and going back into PI. So you can track a bolus of PLC generated DG through that cycle. I don't think there's anything we've done that can rule out, though, that there isn't PLA2 involvement in creating and maintaining the acyl chain composition of each phosphoinositide, because we haven't directly looked at it. So it's possible. I mean, I don't know, Ed, have you ever found any evidence for PLA1 or A2 activities working on PIP2 or PIP? I couldn't rule out that there is some involvement at PLAs.
Let me ask, on the DAG, if you look at the DAGs, which is your precursor, is the arachidonic in the 2 position, and is the steric in the 1 position or the 3 position in the DAGs? Have you looked at those at all? That is that a
so other people have in the past. I don't think you have you directly checked that the 38,4 DAG that gets generated on PLC activation definitely has stearate in SM1 and arachidonate in SM2?
No, we haven't looked directly at that ourselves.
I think it's been done in the past, Ed, but not with our methods and in our experiments.
Yeah, sorry, I think potential issues that people often overlook is that the DAGs are actually very difficult to analyze because they're chemically unstable and you get migration relatively quickly. So when we do analyze DAGs, we find we can't spend very much time with processed samples before they actually degrade. So we have a maximum of about 8 samples that we would run, a batch of 8. And beyond that, we start to see quite serious changes.
That was behind my question, actually, is the sense that that was the case. We haven't looked at DAGs, but we've looked at lysophospholipid like LPC. And this goes back maybe 30 years. Andreas Plukton did it by NMR. If you generate a lyso-PC with the fatty acid on the 1 position, it migrates just about 10-20% to the 2 position very rapidly, extremely rapidly. If you generate a lysolipid with the fatty acid on the 2 position, which you can do with a phospholipase A1, generate it, and then watch its migration, it's extremely fast, and it goes to about a 90-10 mixture. So it reaches equilibrium. So we measured the rates in both directions for lysophospholipids, and they're very quick. And they're catalyzed by acid, by base, by silica gel. If you were to go separate and synthesize them and purify them on silica gel, it'll migrate. And I suspect the same is true for DAGs.
It is. As a chemist, it's very frustrating because we make them in the lab, and if you don't convert them quickly, you get migration.
So Ed, how does the cell protect itself from this happening? Because if it does happen, it just scrambles everything. Well, so are you saying that there's more activity in unscrambling it than we're suggesting? Or yes,
the neutral pH, absolutely neutral pH, buffered as in a cell perhaps, or wherever something's made, is the minimum rate of non-enzymatic catalyzed migration. So depending how fast the next enzyme grabs it or acylates it, it seems to work in cells.
Yeah.
Okay.
It's when you're trying to purify it and study the rates that it's challenging.
Okay. I think that's a great discussion, and we've got several more questions, so I should probably move on to Rosangela Santos.
First, congratulations on your presentation. And my question is, do you think that it's possible to do absolute quantification of these 38 species? When a calibration curve is prepared using endogenous PEEPs, like those ones that are sold by Avanti?
My quick answer to that is yes.
You can. We actually make isotopically labeled 38.4, PIP2 and PIP3 for use with clinical trials, because we found in clinical trial material that gave us the most reliable readout and most accurate readout. If you use something similar and do a calibration curve, yes, it ought to work, and it's probably close enough for most uses. It depends how far you want to go with the accuracy.
Okay,
thank you very much.
Thank you. So Hong Yin.
Okay, I was just curious. I understand that the extraction method you use is acid of modified folk. I think the reason you favor this method is the final extract contains a minimum amount of water, which facilitates the methylation process. Have you tried the Mathias extraction method, which is the methanol methyl tertiary butyl ether? That one is less toxic. has higher extraction efficiency for phosphoinositide?
Yes, we've done that. We find that in our hands, the chloroform methanol extraction media works very well. We haven't seen a benefit to change, but it does work. There have been circumstances where we've needed to use it, and you can. Equally, the main thing is that when you're doing the derivatization, you've got to have methanol present. It doesn't matter if you've got some water. It doesn't matter if you've got any other solvents. The key thing is you've got to have an alcohol. You've got to have methanol, really.
So you're saying the ME
TBDME methanol
it does
mix
work.
should work. Should work, yes. Well, it does in our hands.
Okay, but the methylation efficiency isn't as high as the modified FOC.
I don't think I could say that. I can just say that we have found that the chloroform methanol extraction works really well. We have used the TBDME methanol mix when we're focused on other lipids. So, for instance, we've isolated the CDP intermediates and analyzed those. And I think we use TBDME methanol mix for that.
Yeah,
Okay.
thank you.
Thank you. Miguel?
Yes, I just want to go back a little bit to Ed's question. I guess because we have both spent many years working with PLA2, we are attracted to that part of the thing. And also because I'm calling from Ann Arbor, where Bill Lance showed us his cycle. And the one enzyme that has unmistakable preference, if not exclusivity, for 20 with 4 is MBOT7 or LPIAT. Is there anything known about PLC having any sort of preference to acyl chains?
That's a really good question, right? And it's something I think we were able to look at in the Barnida et al. Paper in EMBO-J that I think is listed somewhere on one of the slides in the presentation. So if you pick a cell line, and I think we used MCF7s, where you have a nice heterogeneous collection of molecular species of PIP2. So this is one that hasn't enriched in 38.4. So you have a nice variety of PIP2s, right? You then stimulate PLC via a GPCR, and then look at the initial rate at which each of those PIP2s is being cleaved. And they are all cleaved at precisely the same relative rate, i.e. they're all cleaved in direct proportion to their starting concentrations. And that, to me, is very good evidence that the PLC doesn't care, right? And it's actually through using that model, we were able to show that it's the 38.4 DG that's made that then is selectively routed back to PI synthesis. So even though all of the different DGs were created equally, the 38.4 as a preference for going back into PI, which suggests that there's acyl chain selectivity at the DG kinase step and subsequently. So I think my answer to your question is I haven't looked at every single PLC. I've only looked at the PLCs that were in that system, which will be some form of PLC betas that were coupled to the GPCR. But at least in that one place where we looked, there really isn't any acyl chain selectivity.
Thanks, Miguel. We've time for one last question from Sheng Gron Lee.
So I just saw your experiment, like you are not able to, you're only able to differentiate PIP and the PIP2 and the PIP3. So you're not able to differentiate on the PIP on the different species, like PIP on the 3 position, on the 4 position, and the 5 position. I'm just wondering if we have like a deuterium labeled PIP and on the different species, but you de-isolate it without the acyl chain, is those head group. with the glycerol on it is like separatable or not? Can be like
Yes.
using
Yeah.
this
So
to
this,
measure.
yeah. So they separate really nicely
Mhm.
by strong normal phase anion exchange chromatography. That's where I started with my PhD and postdoc. Len and I were just building those methods for separating those different differentially phosphorylated head groups where they're phosphorylated in the different positions. You can get those separations to work really well.
So in other words, if I can put the deuterium on the glycerol, you can use that standard to measure do the lipidomics.
Yeah, but well, then you have the problem though of measuring the glycerol phosphoinositides because of all of the polar solvents, etc., and the high iron content. It's not easy to get that to work with the mass spec, is it, Jonathan?
We did look at deacylation for a short period of time, and it didn't look like it was going to be a route that would work well for the mass spec. I can't remember the detail of what went wrong because we did it many years ago. We quickly decided that just shortening the chains through the use of ozonolysis would give us a similar effect. And that's actually the route we then went down.
I think there is a group in the Netherlands that have come up with a method where now when I'm thinking about it, where they almost desalt the eluate coming off the HPLC to get it into a form where it can go into the mass spec. But I think they still end up with overall sensitivity issues compared to the setup that Jonathan has. You know, it might be possible if you really put some time into it. Obviously, you lose the acyl chain information, but if you really focus on just wanting to measure the different positional isomers, probably strong anion exchange chromatography on party-sphere-like columns with phosphate gradients is still the best way of separating those different isomers.
Okay, thank you. I mean, we're slightly over time, but I've got one last question. I think following in the vein of lots of technical questions here, which is great, actually. I think this is really good. The 18O water work that you talked about, that there you're talking about the 18O going onto the phosphate. We've used it in experiments looking at phospholipids, like esterification of fatty acids into phospholipids, because of course, when that happens, you get incorporation of 18O in that way as well. So I'm wondering about the implications of that going on at the same time. I mean, obviously, if you're fragmenting off the head group or the phosphate in the head group, you won't have a +2 on that end, but you may have it on the other end. And so, you know, newly formed phospholipids might have a mass increase of 2, and you may have that may somehow affect what you're measuring a little bit.
Well, I was going to say that's a good point, but we've been going at really early times and we're looking at mass conservation over the times that we've got. So we haven't actually seen it either. That would cause us a problem with our, we always run things through a neutral loss scan, for example, where we're looking at the diacyl glycerol fragment, and we don't see changes in that fragment. So we're not getting O18 incorporation into those esters.
Well, you'd
see the time frame we're looking at. And I think
it depends on the cells. I mean, if you're looking at like neutrophils, which you talked about earlier, and other kinds of immune cells, that sort of remodeling happens really fast when they're activated. But I suppose it all depends on the steady state levels of the different species too. So, you may see an increase in some of those forming at the same time, but you'd have to go and look for the masses. I mean, they're all going to be +2 over for the precursor mass, at least, aren't they? Yeah. I mean, maybe it's such a small proportion compared to what's there that you don't pick it up. But
it was a concern.
We did worry about it.
Okay.
And in the experiments that we were carrying out, it didn't seem to be an issue.
Yeah.
But why don't people use this more? I mean, we started off thinking we were just going to use an isotope log of phosphate and add that to cells. But then that just gets rapidly diluted out in the massive water pool within seconds. So that doesn't work. And then we thought, well, we'll use that to our advantage then and start with the massive water pool.
Yeah.
And look at the penetration of the O18 nuclei into the species we're looking at. And it works really well, Val.
Yeah, it does.
So,
yeah.
you would think that people could start looking at phosphate incorporation into proteins or other metabolites. And yet we really struggle to find anybody that's used it.
Yeah, and obviously we were using it for a completely different
For a
purpose.
completely different reason.
Yeah, just in the same molecules, more or less.
Yeah.
That's funny coincidence, isn't it? Yeah, interesting. Yeah, I don't know. I mean, more ways to look at, lipid flux as well is really needed here. I think that's a different issue to what you're talking about, but yeah, that's particularly in the field that Ed and I work in. Sorry?
Even protein
phosphorylation. Yeah, I don't know. Can't answer for that. I don't work on that, so I've got no insight. Okay, but thank you both for a really excellent talk. It's great to go into details, experimental details. I think is really good, really keen, really makes sure that people are getting support with what are actually really technical experiments, and being able to speak to you guys about the methods you use is so valuable. So thank you very much for that, and thanks to everybody for coming today, and we'll see you next month for our next webinar. Bye.
Thanks for the opportunity. Thanks.
Yeah, bye now.